Grazie, Dario. Good evening. Okay, today I'm going to present to you some of the activities that are ongoing at the Department of Pharmacy, the University of Pisa, regarding, of course, Spoke 4 of the PNR project. So today we are focusing on the main two targets that Dario already talked about, that are pulmonary delivery and crossing of blood-retinal barrier through topical ocular delivery. So these two organs actually look very different, but there are several similar aspects that we can take advantage of for the administration of drugs. In particular, the project and the activities I'm talking about regards different drugs, particularly focused on protein drugs and new molecular entities or new drugs. So for what concerns pulmonary delivery, one of the main aspects among all is the local regional delivery of the drug. So it means that the drugs need to impact on deep lung if we want a lung delivery, or otherwise we can also choose for a bronchi delivery or for upper level of the respiratory tract. And this is either for aerosols or for powders. So within the Spoke 4, it was acquired and installed a big apparatus that is an NGI, is a new generation impactor that is coupled to a breathing simulator. This is a very important instrument for pulmonary drug delivery, and it is optimal and needed for a high TRL level, because it is a pharmacopoeia instrument, but in this special case, it can also be applied for the development of pulmonary inhalation medicines. So why it is important? Is it important because the breathing simulators allow to design and to stratify the population target for the drug in order to have an optimal dosage and to adjust the feature of the nanomedicine or drug that is under development in order to have the highest efficacy. And this instrument, it is applied for the research within the Spoke 4, but as you can see, it is also applied in collaboration with other spokes that belong to the project. In particular, tomorrow, Professor Batoni from Spoke 7 will present some of the results we obtain on the antimicrobial delivery to the lung. So talking about the use of this instrument, I would like to show you how the breathing profile can change the impact of a model protein like BSA or other protein like PLG, that stands for plasminogen, according to the profile that is applied. In here we have normal breathing, and we have normal breathing, or we have acute respiratory distress syndrome under mild, moderate, or severe breathing profiles. So it changes the dose that is delivered, it changes the fine particle fraction, that is actually the fraction that reach the profound lung, and also the aerodynamic medium diameter. So among all these combination that we investigated, I would like to show you how not only the protein and the profile, but also the presence of an excipient can change the dose that is actually arriving to a patient. And in this case, the use of hydroxypropyl beta-cyclodextrin helped to the attainment of the best aerodynamic particle size distribution. And this is important because the cyclodextrins are protective agents that can also enhance the stabilization of the protein, but also the permeation of the protein, especially if we consider that from literature, only the 2 to 5% of high molecular weight proteins, for example, antibodies, can permeate the lung epithelium. So hydroxypropyl beta-sarcodextrin is not currently in use in any medicine for inhalation, but it has been used in clinical trials, so testing the safety of this molecule and its application. So in a paper that belongs to the activities performed within this spoke, we try to enhance the features of the hydroxypropyl beta-sarcodextrin, including also thiol moieties. Thiol moieties can increase the mucoadhesion, so the interaction with the mucins that are present on the surface of the lung, of the alveolar space. And also, as you can see, the hydroxypropyl cyclodextrin, and in particular the thiolated one, are capable to form aggregates. In literature, these aggregates of cyclodextrin are known since a long time, but the problem is that these aggregates are concentration dependent and they are not easy to be stabilized. So they were actually stabilized in this work with a bifunctional cross-linker using small PEG moieties, like 2 or 3 in length. And by arranging the concentration and the reaction condition, it was possible to obtain a stabilized nanoaggregate of thiolated cyclodextrin, so we call them nanoparticles, with an average distribution of 400 nanometers. So these particles were tested and compared to the pristine cyclodextrin with using a model drug desamethasone that is very common and a very labile sensitive vegetal extract. They were tested on an in vitro air liquid model that is an in vitro lung model. And in this particular application, we place the cellular monolayer in the second stage of a twin stage impinger. That is an instrument where in this part we can see the pulmonary section and this the bronchi section. So the insert is placed in here, so we can test exactly the drug that, or simulate the drug that would impact in vivo on the lung tissue. And cytotoxicity, permeation, and biological activities were performed. For what concern the behavior of these nanoparticles based on thiolated cyclodextrins, we see that they are capable to increase the permeation of the drug across the lung simulated monolayer. And also they can protect the labile molecules, and we can have a biological activity that is enhanced thanks to its encapsulation. Moving toward, in collaboration with Professor Lisanuti and Professor Tagliani, we would like to focus on non-small cell lung cancer that is a very aggressive pathology and unfortunately very common. And the use of gefitinib that is already used in clinic, but is with an oral application leading to several side effects. And in this case, we are working with a methylated beta-sarcodextrin, also thiolated. And why it is like that? Because we found out that this kind of cyclodextrin is forming very, I would say, tiny nanoaggregates with an average diameter of 1.5 nanometers. So we are now working on the stabilization of these nanoaggregates, and the work, of course, is on progress, but by now we also are capable to still obtain hundreds of nanometer aggregates. So we would like to push a little bit more on the very tiny nanoscale. For what concerns the encapsulation of gefitinib, it is this drug has a very nice affinity with the cyclodextrin with the association constant of 350. And the work is proceeding also in collaboration with the Scuola Normale Superiore, Professor Cardarelli, because gefitinib has a nice fluorescent properties that are studied with fluorescence lifetime assessment. For what concerns the eye application, this work is performed by the group of Professor Patrizia Chetoni. It is a focus on a nano-assembly system for ocular delivery of proteins. And the study is focused on the use of poloxamer of different type, forming polymeric nanomicelles in presence of vitamin E derivative, that is TPGS. And the study involves several steps: in vitro preparation, characterization, diameter distribution, absorbance, fluorescence, rheology, and so on. And today we can show you that these micelles, the best that were selected are actually the mix PX338 and the mix PX407 that have, the one with the lowest size and with the lowest also critical micellar concentration. So these selected compounds, the selected micelles were loaded with bovine serum albumin as model protein, and the biocompatibility was assessed in vitro on rabbit corneal epithelial cell lines, showing a very nice biocompatibility with a cell viability that is up to 80% for the concentration tested. And also the release of the drug from these micelles is evidencing that in the case of mix PX338, we have a faster diffusion of the protein. Sorry, I always click the wrong button. A fast release of the protein says that the nanomicellar structure effectively can enhance the drug release, whereas in the other case that the protein is more retained within the micelles. To conclude, we also are investigating some other nanoparticles for ocular application, strong of a past deep investigation of the application of chitosan derivatives for nanoparticles for ocular administration. And in this case, we are still in collaboration with Professor Nuti and Professor Tagliani. We are preparing a nanoparticle loaded with the model drug bovine serum albumin. These are composed of 3 different chitosan derivatives. One is a pristine chitosan, the other one is a quaternary chitosan, and the other is a sulfobutyl chitosan, in order to have different charges on the surface. And these are under evaluation in blood retinal bioreactors in collaboration with Professor Vozzi. The coordinator of the activities at the Department of Pharmacy is Professor Calderone, that is a pharmacologist. So the activities that will be performed by Professor Calderone in collaboration with Dr. Maria Franzini, they regard the cytocompatibility aspects and the pharmaco distribution. So first, the evaluation of safety and efficacy will be performed in cytocompatibility test and hemocompatibility test according to standard indications. Selecting the cells that are involved in the model that we are studying and applying them to the drug efficacy endpoint. For example, in the case of tumor application for anti-tumoral efficacy. Within the spoke, it has also been acquired another instrument that is a flow cytometry. That is a very powerful instrument that can separate cells either if they are in suspension or before they were cultivated in plates in adhesion. And they can be separated depending on the fluorescent labels that they have. So they can help in the evaluation of safety, efficacy, and targeting. We are also investigating, the application of this instrument for the separation of labeled nanoparticles, because the instrument have a sensibility that goes under to 100 nanometer. So it is possible that for particles that are in this range, we can apply the instrument also for the separation of particles that are labeled by different flags. So for what concerns the pharmacokinetics, it will be assessed in vitro 3D models using different type of human tissues. And in the end, we can also think of an in vivo pharmacokinetics on rodents. So that's all.